After the centrifuge stage, the plasma samples should look like this in the purple top tubes, and now we're going to see the extraction of the serum from the red top tubes. So Amanda's removing the top, and here are the pipette tips.
She is aliquoting 150 microliters into serum tubes 1 through 6.
So I start at the top and suck it up into the tube slowly, making sure not to stick my pipette tip down far into the serum and not to touch the red blood cell. And take the tip, put it in the tube, and push down past the first clip to get all of the serum out of the pipette. Okay, I completed aliquoting the first 6 tubes with 150 microliters in each. The next 4 will have 250, and all of the remainder after that will have 500. So I put my tubes in the ice bucket with 1 through 6 in the first row, 7 through 10 in the second row, and 11 to 25 in the third row for a physical reminder to change my pipette.
We're working on the plasma and the buffy coat part of the protocol. The plasma bucket is set up exactly how the serum is, with the tubes in order from 1 to 6, 7 to 10, and 11 and up, aliquoting the same exact amounts into each of those tubes. So now we have our buffy coat tubes. Plasma tubes, after they spin down, you will see that you have a top layer, which is the plasma, a middle thin white layer, which is the buffy coat, and the bottom layer, which is the red blood cells. So now I'm going to take the top, which is the plasma, and aliquot it, the whole thing, into the plasma tube here. Then I will take the same transfer pipette and suck off the buffy coat and stick it in the bar-coded labeled tube labeled buffy coat. So I'm going to go ahead and do that now. I will take my transfer pipette provided in the kit, and you want to suck off on the top, nice and easy and slow, so you do not disturb the white buffy coat layer. And you want to leave about a 1/4 inch to the buffy coat so you do not disturb it. So now I'm going to take this into our plasma, and I'm going to get the remainder as much as I can without disturbing the buffy coat. This is about good where you can stop collecting the plasma, and now I'm going to collect the buffy coat. This is the most important step of the protocol, because if you do not get all of the buffy coat, then we can't get any DNA from this person. What you're going to do is take your transfer pipette, stick it on the top, and you're going to suck the entire white layer up, including the red blood cells. This is very, very important. Then you're going to take this entire amount and put it in the bar-coded label tube, labeled buffy coat. And you want to get rid of your transfer pipette into 10% bleach. You want to let it sit for 10 minutes before you dispose of it in the biohazard waste. And you will repeat the same exact thing for the second tube. And you have these 2 tubes. This is buffy coat 2, and you will have also another tube for plasma. This is the plasma we just collected, and we are going to centrifuge it for the 5-minute step. It is listed in the protocol. So I'm going to go ahead and put it in the centrifuge, and then immediately after I'm going to aliquot it the same as I did the serum. Whenever you spin this down, you'll get a tiny little white pellet at the bottom. That's just getting rid of any other cellular debris that we do not want for proteomics. And you aliquot it cap everything and put it in the freezer and make sure you mark down the freeze time. And that's it. Buffy coats go. Always make sure you have caps on your centrifuge, especially for the glass tubes in case they break. And that is our centrifuge. This is the setting for our centrifuge here, 1500 G for 5 minutes at room temperature.